So this is just kind of review what we did yesterday. What is this right here?
Axon.
It's a neuron, also called a nerve cell. What are these called?
Dendrites.
What's that called?
Cell body.
What's this long thing called?
Axon.
Axon. What are these things called?
Myelin sheath.
Schwann cells that form the myelin sheath.
Oh.
And what are these little areas between the Schwann cells called?
Node of Ranvier.
That was so good.
Oh, yeah.
How does an action potential occur? When a neuron is not transmitting a signal, there is a separation of charge across its membrane. This is called the resting potential.
So, I'll do the talking here. It's a lot more interesting than that guy. So what happens is you got sodiums on the outside, potassiums on the inside, and some negatively charged ions on the inside, that overall make the inside of the axon have a lot lower charge than the outside of the axon. And they show that to you in this diagram by putting a plus on the outside and a minus on the inside. As a signal moves down the axon, this is what happens. Here comes the signal. We call that what? Do you remember?
Active signal. Action potential.
That's the action potential. As the action potential comes down, little channels in the membrane here. There's channels in the membrane. They open up and let sodium come in.
When I was reading, it said that like sometimes it does the things where it does it channels, and then sometimes it doesn't. Or maybe I read it wrong.
I don't know about that.
Probably.
Anyway, when the sodiums come in, it's positively charged on the inside. But that's just a brief, because the potassiums are going to go back out, and it'll be negative. It'll be positive again on the outside and negative on the inside. So it's just a brief pulse of positive charge that's on the inside. That positive charge. Oh,
sorry.
Question?
Oh, so when the sodium's in there with potassium, it's positive instead.
That's right, because sodiums are positively charged, and so it raises the amount of positive charges on the inside of the atom.
Potassiums are also positive.
Potassiums are also positively charged, but the potassiums there aren't enough of them to overcome the negative charge proteins that are on the inside. So overall, the charge inside is a lot less than the charge outside in this case. But when the sodiums come in, now the charge on the inside is much greater because the sodiums are positively charged, and now you have an overall positive on the inside.
Isn't that called something with like a D, like
Diffusion?
Depolarization?
Yeah. Oh, depolarization, yeah. Depolarization, we say that the membrane is depolarized. Yeah, good. Yes?
Actually, in the video, it said something about the reasons that they're going in and out has something to do with the concentration gradient. What is that?
Yeah, okay, look right here. Sodiums are in high concentration on the outside,
Yeah.
and they're in low concentration on the inside.
So you always want to
So when those channels open, they're going to come right in.
Okay.
That's just simple diffusion, and that's what we call the concentration gradient.
Do negatively charged protons come out when the sodium goes in?
No, they don't. None of the negatively charged proteins come out. So really, it's not negative on the outside. It's just a lot more positive on the inside. And so they show you that by putting a negative here, but that's not technically correct. This would probably be somewhat positively charged, because all the sodiums don't go in. A lot of them do, though. So in reality, there would still be a few sodiums out here. Are we good?
And how does this only happen in the node part?
In the axon, you mean?
Well, you said this only happens in the node.
That's a good question. It doesn't happen where the myelin sheath is. I'm not sure why that is. I don't know.
What's the point?
Well, what happens
This
when
is
the
an
myelin
axon.
sheath is?
I don't know.
No one?
Somebody might, but I don't. Somehow the signal seems to jump from node to node. Like, this will happen here, and then it'll happen here, and then it'll happen here, and I don't know how it gets transmitted from here to here, Somehow the myelin carries that charge and causes the next channels to open, but I don't know how it does
So
it.
it says saltatory conduction, right? Is that what it?
Saltatory conduction means hopping, going
Yeah.
boop, 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 boop.
So is that how it crosses?
Well, I don't know what is happening to allow the signal to get from here to there. Somehow it does, but I don't know what it is. Maybe somebody does, but they aren't going to ask you that on the AP test, so we shouldn't worry about it.
Neuroscience is fascinating.
Yeah. So, the sodiums come in, the potassiums go out, and the next one down, the sodiums come in, the potassiums go out, and the next one down, the sodiums come in, and the potassiums go out. And that just keeps going, and the signal moves down from left to right, or, from the cell body down the axon toward the end. And now, are we back to where we started?
No, sodium potassium pump.
Sodium potassium pump that's also in the membrane here has to switch them all back. This is my favorite part of this video where they switch back. Whoop. They switch back. Forgive me, I've watched the video several hundred times now. Okay. So, now,
Synapse.
so yeah, it jumps from node to node somehow. That's saltatory conduction.
Just like saltation geology.
Yeah, I'm not sure what the root of that means, but that's...
Saltar
Means
means
the
to jump in
hopper to
Spanish.
jump.
Does it?
It's mentioned that.
Saltar means jump?
Or something like that.
It also happens at the bottom of rivers when water's going over stones.
Yeah, that's right, it does.
That was a science-full question, I guess.
Yeah. So here comes the signal down to the end of the axon. This is called the axon bulb. And the end of the axon has little vesicles. These are little bubbles. Y'all know what vesicles are. That contain special chemicals called neurotransmitters. When the action potential reaches the bulb, the neurotransmitters are released by exocytosis. Do y'all remember that process? And the neurotransmitters now will cross a small space that we call the synapse and will bind with receptors on the next protein, on the next neuron. And when it binds, when this little protein here binds, it has to have exactly the right shape. But this protein has exactly the right shape to pick it up. So when that thing binds with that thing, when the neurotransmitter binds with the receptor protein, it causes what we call a conformational change, a shape change in this protein. And that protein opens up, and guess what's coming in right here? Guess what those two green balls represent? No, not neurotransmitters, that's the neurotransmitter. It's some ions that we just talked about.
Potassium.
Not potassium, what goes in?
Calcium.
Sodium goes in, potassium goes out, and a new signal gets started on the next neuron. And Hannah, that's where the signals comes from. You asked that yesterday. Remember I said you're gonna have to wait? And you got real mad at me. I'm kidding, you didn't. Yeah. Well, she had her hand up.
No, go ahead. Put your hand up. Go ahead. Go. What kind of changes did you say it was? Is it something like a seed?
A conformational change means the shape changes when it receives that neurotransmitter. What were you going to ask, EKS?
Okay, so is the signal in the neurotransmitters that are excreted?
Well, this signal has come along
Yes.
and caused, somehow when the signal comes along, these bubbles that contain neurotransmitters, they're released. And so the neurotransmitter is released into this space, and those molecules will just float around until they happen to bind with this protein receptor.
Okay.
And once they bind with this protein receptor, sodium will then rush in.
Is that a gate?
This is a sodium channel, that's correct. And it allows sodium to rush in, and potassium will go out, and we'll start a new signal in the next neuron. So this is how neurons communicate with one another, which is what the AP biology test thinks is real important. Communication is one of their big things. Yeah.
What's the difference in synapse and is a synapse in the axon?
A synapse is this connection between this axon and this dendrite.
What's the bulb?
That's called the axon bulb.
No, what's the synapse?
The synapse is this, I think that the synapse is actually the space right here.
Then what's the synaptic cleft?
The synaptic cleft is the space there. I guess the synapse then would be the whole connection between the two. This whole thing. How that one axon connects with a dendrite or a cell body of another nerve cell. That whole connection is called a synapse. They often say that the nerves synapse together, or there's a connection as a synapse.
Is there something else where the calcium enters?
Yes, if you want to get technical about it and look at what's happening up here, when this signal comes down, it causes little calcium channels to open in this membrane. When the calcium rushes in, it attaches to a carrier molecule that moves the vesicles to the edge. That's technically what's going on inside this axon here. And I have another video that shows that if you want to see it.
Is the protein that locks on to the gate, that's the potassium?
That's a neurotransmitter.
And then potassium goes in?
Sodium goes in.
Oh,
Potassium comes out.
okay.
To get a signal going, it's sodium coming in, potassium going out. Watch this little video here.
Synaptic terminal cause voltage-gated calcium ion channels to open.
Oh no, let me start it over.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Acetylcholine molecules diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium ion channels. This causes the ligand-gated sodium ion channels to open, and sodium ions diffuse into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
So how far is the space between the two things?
Micrometers. The space that these neurotransmitter molecules are crossing is very tiny. Acetylcholine is the name of one type of neurotransmitter. There's a bunch of different types. There's one called serotonin. There's one called dopamine. You ever heard of dopamine?
Yeah, dopamine.
That's released in your pleasure centers of your brain. That's why they used to call smoking marijuana smoking dope, because it releases dopamine. It causes dopamine to be released, which activates your pleasure centers of your brain. Yeah.
What's the receptor called? Is it just called a protein receptor?
It's just a protein receptor.
Okay, well, what was the other thing you were saying?
I don't know.
It wasn't the bulb, it was something else. Never mind.
I say a lot of things. Okay? Are we good?
So how does the brain work?
By the way, we call these ligand-gated sodium ion channels, these things. A ligand is a molecule that is received. So that's technically these acetylcholine molecules are ligands. A ligand is kind of a general term. I don't know if you've ever heard that word before. And so this will not open without the ligand attaching to it. That's a word that may come up on the AP exam. I don't know. Ligand. A ligand is required to open the gate. So they call it a ligand-gated sodium ion channel. So, you have set signals coming in to the nerve cells, and it's the communication between nerve cells that is going on in your brain when you're thinking. This is all very important in getting signals to go. This video right here, it's kind of boring, and the guy speaks very monotone, so you could get real bored with it and just decide you're going to go to sleep. But it's good information, so see if you can follow this. It talks about how these signals work together.
Nerve cells, or neurons, are the basic units of the nervous system. Neurons serve as communication lines between other cells.
Watch it, Sarah.
Their long processes conduct electrical signals that can be passed from cell to cell. Neurons vary in shape and size, but they generally have three main regions with different functions. First, dendrites receive signals from other neurons and conduct signals toward the cell body. Second, the cell body integrates the incoming nerve signals. And third, the axon conducts electrical signals away from the cell body to other cells. Axons are frequently surrounded by a myelin sheath that helps signals travel very rapidly, sometimes reaching speeds of more than 100 meters per second. The long axon ends in several short branches tipped with enlargements called axon terminals.
Pay attention here.
The junction where the axon terminal from one cell meets another cell is called the synapse. In almost all synapses, transmission of signals is in one direction only, from the axon of the first, or presynaptic cell, to the second, or postsynaptic cell. At the synapse, a chemical substance called a neurotransmitter is secreted from the end of the axon terminal. The neurotransmitter carries the signal from the presynaptic cell to the postsynaptic cell. Axons from several neurons can make contact with one postsynaptic cell. These synapses can be either excitatory or inhibitory. The postsynaptic cell integrates incoming signals from several cells in the following way. Often, signals from many neurons are required before the postsynaptic cell can relay a signal. Receiving a signal from an excitatory neuron causes the postsynaptic cell to more easily relay the signal down its axon. The effect is additive. The more excitatory signals a postsynaptic cell receives, the more readily it sends the relayed signal or signals down its axon.
Okay, let me pause it for a minute and tell you what's happening. The pink ones are excitatory. So they send out neurotransmitters that causes the channels to open and send a new signal.
What are those things?
These?
Yeah.
That's the end of axons,
Okay.
where the synapses
Those
are.
are terminal.
What do the excitatory ones do?
So the pink ones are excitatory. They cause this neuron to send a signal out. The blue ones are inhibitory. They cause this neuron to not send the signal out. Savannah, you getting all that? So there's a combination going on, you see here. There's excitatory signals coming in and inhibitory signals coming in. And it's the combination of the two that causes where this cell determines if a new signal will be sent. If it's getting a lot more excitatory signals coming in than inhibitory signals coming in, a signal will be sent. But if it's getting a lot of inhibitory signals coming in, it's not going to send a signal.
Receiving a signal from an inhibitory neuron hyperpolarizes the postsynaptic cell membrane, causing it to resist transmission of a signal down its axon. We have indicated this change in membrane permeability as the glow surrounding the cell body. The inhibitory effect is also additive. The more inhibitory signals a postsynaptic cell receives, the less able it is to send signals. Excitatory and inhibitory signals counteract one another. A larger number of inhibitory signals will block the transmission of a few excitatory signals. In addition, an inhibitory synapse close to the axon can block the effects of signals from excitatory synapses farther away.
Okay, so what he's saying there is, if the signal is coming in close to the axon, it has more of an effect than a signal coming in far away from the axon.
Similarly, an excitatory synapse close to the axon can override an inhibitory synapse farther away.
Now, in your brain, one neuron might have 10,000 other neurons connected to it. And so all these signals are coming in, and it's this, and every neuron has that going on. So it's a very complicated system. And that's why it's been so hard to figure out how people think and how memory is stored, because it's so complicated. Yes?
What were they saying about glowing?
They were saying an inhibitory signal
Makes the whole
coming in, an inhibitory signal coming in will cause the neuron not to send a signal, and they'll show you that by making the thing glow. It doesn't really glow. Yeah.
Good.
So the ones that are, what was that about distance, like the farther away?
So this is an excitatory one, and it's near the axon. It would have more power than an excitatory one far away.
So the closer, the more effective.
The closer, the more effective. Yes.
How do they study these things?
Well, they started studying these things with the squid axon because it's real big. And the reason why a squid axon is big is because it doesn't have any myelin on it. Invertebrates never evolved myelin. So the only way they can get their signal to go faster is to grow neurons bigger. So squid neurons are huge. And you can actually see them. And so that's how they started studying these things, was with giant squid axons. And they stick electrodes in there. Yeah. Well, they do surgery on the squid. They put it to sleep. And put little electrode here, and activate the neuron. And they stick little sensors inside the neuron to see how the things were changing. Nowadays, they have better equipment, and they can actually work with like a rat neuron. They can actually work with that because they have such better equipment. But it's interesting how they do this work. That's all neurology, the science of studying nerves. Yeah.
Isn't it possible for people to have a disorder where they can't feel pain?
Yes, that's correct.
Awesome.
And that's not good because you don't know when your body's been damaged. People who can't feel pain usually lose their arms and legs in some way. Like they'll put their hand on a hot stove and they won't feel the pain, so it ends up catching their hand on fire or killing all the cells there.
Can't they feel their hand like getting warmer or something?
They can't feel like
Perhaps, I don't know. It might depend on the person, but you cut your leg, you don't feel it, you bleed to death. That's not a good thing.
Yeah, so where do psychology and neuroscience meet? I mean,
they're very intertwined.
Yeah.
So if you take psychology, we talk about a little neuroscience in every chapter.
Yeah. Is it really known or known in some detail?
I mean, the only reason you think is because of the neuron activity in your brain, so it's completely intertwined.
Yeah.
Okay, so the next thing we need to learn are the parts of the brain. And the brain is just a giant collection of neurons. And the brain actually comes in two halves.
Hemispheres.
Two hemispheres. You have your left hemisphere and your right hemisphere. And they both have the same parts to them. So there's a redundancy there. In other words, if something screws up, doesn't work over here, the other side of the brain can take over for it to some extent, which is nice. For the most part, the left half of the brain controls the right half of the body. The right half of the brain controls the left half of the body. There's a crossover there. And I'm not sure why that evolved that way, but it did.
Because my grandma had a stroke on the left side of her brain, so the right side of her body.
Good move, yeah. And the brain is divided up into parts. This big part here, this is the cerebrum. And it does all your thinking and memory and problem solving and personality. Everything you're consciously aware of is here in the cerebrum. It's huge in humans. It's a lot smaller in all other organisms. We've kind of had a runaway brain growth.
Steroids.
And it caused us to be able to manipulate the environment. Man, we can just do just about anything, can't we? This part of the brain down here is called the brain stem. It was the first part of the brain to evolve. You see that in the earlier animals. Reptiles and fish have a brain stem, but they don't have much of a cerebrum. That controls, the brain stem controls all your automatic functions. And then this part right here is called the cerebellum. It controls muscle coordination. It's hard to coordinate the movements of all your muscles, Jonathan. Difficult. So you have a whole big part of the brain just there to coordinate muscle movement.
When you say automatic functions, you mean like breathing?
Breathing, heart rate, your emotions, fear, anger, love. It's all right here in the brainstem. Hunger, thirst, blood pressure, controlling your
Sweat glands.
body temperature control. It's all right here in the brainstem. So the brainstem actually has some different parts. You got the midbrain up here. There's a lot of reflexes with your eyes associated with the midbrain. The pons is right here. That's kind of a relay station. Signals come up to the pons and they're sent to different parts of the brain. The medulla oblongata right here. The medulla controls breathing and heart rate, along with the pons helps out in breathing. So breathing and heart rate are controlled right there. If those are damaged, you'll quit breathing. But it's buried real deep in the skull, so it's hard to get to those. This part up here is called the diencephalon. It's got the hypothalamus. That's an important part. That controls hunger and thirst, and it controls the pituitary gland that sends out all the hormones. The hypothalamus also controls body temperature and blood pressure.
What is the midbrain?
The midbrain is kind of a relay station, a reflex center. Signals will come in there and go right back out to your eyes for one thing. It helps with your eye movements. You have a lot of reflexive eye movements that you do without even trying. If there's a bright flash over here, your eyes automatically move toward it without you even trying. You don't have to think about it. It just goes straight to the midbrain and back. There are also reflexes associated with hearing, so you can move your head. Have you ever gone up to a dog and gone, and it goes like this?
Mm-hmm.
It's trying to figure out where that sound is coming from, and by moving, changing the position of your head, it can figure that out. That's a reflex. It does it without having to think about it. Yeah?
Okay, so in the diencephalon, it holds the hypothalamus and the what?
The diencephalon has the hypothalamus. It also has the thalamus, which is another relay station.
Oh no, I was talking about the pituitary gland.
The pituitary gland is a gland that produces, it's a master gland of your body. It produces a whole bunch of hormones. We talk about that in 3 chapters.
The cerebellum, you said it controls everything you don't know you're doing, but isn't that the same thing?
No, the cerebellum is muscle coordination.
No, cerebrum, sorry.
The cerebrum is all your consciousness. So cerebrum is conscious activity. So you think, I'm thinking right now, I'm thinking with my cerebrum. I store memories in my cerebrum. Organisms without a big cerebrum, they don't remember stuff very well. You can't teach a goldfish many tricks. It doesn't have much of a memory.
Why do we forget things? There's like not really.
That's one of the big questions of psychology. Why we forget.
Why can't we remember when we were like 1?
Scientists don't really know, but they know these brain cells die over time. And if you, the more brain cells you lose, the worse your memory gets. Why don't we remember stuff from where we're kids? That's called childhood amnesia, and nobody knows why that happens either. We study it a little bit in psychology, but the answer is nobody knows yet. They think maybe you're coding differently when you get older than you do when you're a kid, and you stop using the same coding signals. It'd be like switching from English to Spanish. And once you learn the Spanish well enough, you don't remember the English, so you can't pull those English stored memories out, only the Spanish stored memories. But it's a little bit different. That's a hypothesis, though. That's not proven. They think the way it's codes might work differently. This area right here is called the corpus callosum. It connects the two halves of the brain together. So the two halves of the brain can speak to one another, can send signals back and forth through the corpus callosum. Sometimes they'll cut the corpus callosum surgically to keep people from having real bad seizures. And then the two halves of the brain cannot communicate with one another very well. And there's some real weird psychology experiments where these people, for instance, they can if you separate, if you have them look straight at a screen and you show a picture of one guy over here and a picture of a little girl over here, this side of the brain will see the guy, this side of the brain will see the girl, and they won't be able to communicate with one another. So if you say point with your left hand to what you saw, the left hand will point to the guy, and point with your right hand to what you saw, the right hand will point to the girl. And the guy won't know what's going on. That's cool psychology stuff. Take a psychology class. Anyway, keep reading 37.3.